So we should be live now. So hello, everyone. It's nice to be here back after this summer break. I hope everyone is fine. So I'm very glad to start today with Kevin Bender, our speaker. So I give, as usual, a very brief introduction, and then Kevin, the stage is yours. So Kevin received his PhD at UCSD. Where he worked with Dan Feldman on understanding the cellular mechanism of cortical plasticity. Afterwards, he moved for his postdoctoral training to Portland, where he worked at the Oregon Health and Science University, and where he was studying the cellular mechanism involved in initiating and modulating the neuronal output. So currently, Kevin is an associate professor, if I'm right, at UCSF and in the Department of Neurology. Very nicely, maybe also important for this crowd, is also very heavily involved in the neuroscience graduate program over there, which I think is pretty nice. And overall, his lab is interested in studying neuromodulators in both in health and disorders, and that's what we are probably today hearing more about, the disorder state. So once again, Kevin, I'm very happy to have you here. I hope so. Someone is writing that they don't see anything. But maybe someone, I mean, okay, so others can see, so maybe it's not a problem, so it should be fine. So it's working for others, so that's okay. So again, thank you very much for joining, and now you can share your screen, and hopefully everything is be fine. Otherwise, don't worry, I'm here. So
okay, I'm going to get verification from you, Gaia, that that everything actually gets shared.
Yes, just go ahead.
Properly. Is that good?
So not yet.
Oh.
Now we have a black screen.
Okay, that's so, progress.
Yes, I try to close the down. Now stop. Now it works. So just you have just to go in and.
Hold on 1 second. I'm going to try one other approach. I'm going to share the entire screen in just the. All right, now we're going to have.
That works. That's good. So just go in your presentation mode, and then that's perfect. So here you go. Stage is yours. Just get started.
Okay, great. Thank you so much, Gaia, for having me in my little office in the smoke-filled world of California. It was lovely to hear that other parts of the world have actually been doing things correctly with COVID, and you actually have school, and the kids can go to school and all that fun stuff. It's a little bit different in California right now, but we're making the best of it. And honestly, it's kind of fun to be home. Yeah, so we'll get started with it right out of the bed. It's about 8 a.m. here for us. I want to tell you this morning or this afternoon or this evening about the work that's been going on in the lab with sodium channel dysfunction in neurodevelopmental disorders. And this is work that, hold on, I'm going to bring up a little laser pointer here. This is work that's been done by a trio of people in the lab, and not actually in my lab. Roy Ben Shalom, who is a postdoc in the lab, who's done a lot of computational work and biophysical modeling. Perry Spratt, who actually just defended his thesis virtually and has now moved on to bigger and brighter things, did a lot of the slice work here. And they worked with Serena Tamura, who's a graduate student in Nadav Ahituv's lab, working on some translational aspects of what I'll tell you about today, which focuses on one particular sodium channel, and that is called SCN2A or NAV1.2. So SCN2A is this gene that's probably known fairly well in the epilepsy field because it's associated with a lot of different early childhood epilepsies, some of which are quite severe. But also it's gained recognition in the autism field due to its function or its prevalence in autism genetics. But it's a little bit of an outlier. So it's a strange gene when you look at the genes associated with autism. There are a lot in terms of high confidence autism associated genes that are associated with synapses, and there are a lot that are associated with chromatin, and Gaia's lab does a heck of a lot studying these sort of associations. And then there's us who have been studying this outlier, this ugly duckling SCN2A, which is generally not those that are associated with chromatin modifications or associated with synaptic function. It's actually a gene that encodes the sodium channel that's typically thought of as being involved in firing of action potentials and the initiation of action potentials in the axon initial segment. But we kind of have to study it. And the reason we have to study it is that if you look at the autism genes, it's actually one of the most prominent in terms of the numbers that you see coming back from genome or exome-wide sequencing. And so if you look at the number of protein truncating cases that are associated with ASD within a sample of about 12,000 ASD cases, this is early work from Spark and Autism Sequencing Consortium, you see that it's really high up there, but it's also high up there in terms of missense variants, and those are those that alter the channel but don't destroy the channel. It's way up there with other genes that we all know, like CHD8 and YAP1, anchorin-2. Interestingly, this association just gets more and more pronounced as you increase the numbers in your exome-wide screens. And so over the course of 2 years, instead of us adding an N of 2 or 3 over the course of a day, these guys added another 8,000 cases. And under this case, under total samples of about 60,000, you can see that SCN2A is far and away has one of the strongest associations. So even though it's this odd channel that's associated with something not typically thought of in terms of autism, whether it be synaptic modification or chromatin modification, there's something about it that sort of demands that we investigate it further, okay? And so this actually opens up a situation where, and as I mentioned before, this is a gene that's not only associated with autism, but it's actually more commonly associated with early childhood epilepsies. And there are actually 2 different types. There's either something called benign infantile seizure, which is very early onset, usually within the first couple of months. Magically, this resolves by about a year or 2 of age. And that's a wonderful thing to see happen, and just spontaneously resolves. There are other forms of epileptic encephalopathies that are extremely severe and unfortunately do not resolve with age, and these are very hard to treat with anti-epileptics. And then on the flip side, there is autism spectrum disorder. It is typically comorbid with intellectual disability, but the thing that's interesting about those children is they actually they do not develop seizure very early in life within the first year, but some of them develop seizures later in life. And it looks like it's about an equal number of those that develop seizure later in life as those that develop epileptic encephalopathy, even though if you look at the estimated incidence, the ASD cases probably should outweigh those that are associated with epileptic encephalopathy by about 5 to 1. And so that suggests that there's a population in the ASD field that never develops seizure, and then there's some that do develop seizure. This is something that's really interesting, how changes in a sodium channel that are associated with ASD give rise to seizure. And this is something I'll talk about in a little bit. Okay, but overall, we estimate that worldwide, you're probably seeing about 7,000 new cases of some SCN2A associated disorder per year. And so it's very prevalent. It's one of the most common genetic disorders in children, right up there with Dravet syndrome in terms of incidence. And so it's something that we really need to understand in terms of how do you have these different situations where either you can develop seizure or you can develop intellectual disability, autism spectrum disorder, and what gives rise to these different phenotypes, okay? And so there's actually a unique opportunity here, and that is that this is a really nice gene to study in terms of autism, and that's because, well, we know a lot about its genetics. I already showed you within exome sequencing, we have the highest prevalence in SCN2A, and we know it precisely where individual variants are mapping within the gene. We know what this gene does because it encodes a sodium channel, and the field of neuroscience has been studying sodium channels. We can say we're close to 100 years now in terms of studying sodium channel function. And the nice thing is once you look at those sodium channels, which you can assay very rigorously and quantitatively with biophysical studies, you can then look at them and look at their function within brain cells and circuits, look at animal models that have been affected with either gain of function or loss of function of the sodium channel. Ultimately, our goal within the lab is to translate that understanding into some sort of help for children with SCN2A disorders, and hopefully translate going all the way from a gene into future therapeutics for treatment of these sort of things. Okay, so within our lab, we've actually focused, or we've gone pretty broad in terms of our focus. And we've asked a bunch of different questions. The first is, how can dysfunction in one gene give rise to these different disorders, epilepsy, early epilepsy, or later epilepsy with autism spectrum disorder? How are the circuits and cells affected when you have individual variants in SCN2A that give rise to changes in your sodium channel complement? And then finally, can we leverage that to help families? Okay, so let's focus on the first one. First one is, how do you have one gene lead to these very different disorders? And so the way to do that is to first look at the genetics and look at where different variants associated with different disorders find themselves on the gene. And so this is the encoded gene, NAV1.2, in a protein, and this protein is now flayed out like a reverse sushi California roll on your membrane. And so normally it's packaged like this, and it has a sodium selectivity filter in the center of the channel, and all of these subunits and transmembrane domains are packaged around it. Some of the most important areas here are the fourth transmembrane domain, because that's the voltage sensor. And then also these black dots, DEKA, these encode the sodium selectivity filter, which makes this channel selective for sodium and basically no other ion. Okay, so first thing to do is just look at where different variants are associated with disorders find themselves on this channel. And so if you screen all of the literature and look for those associated with benign infantile seizure, you'll find that there are actually a lot that are near or around the voltage sensor. And this makes sense because this is associated with epilepsy. And one of the common things that you find with epilepsy, with any gene, any voltage-dependent channel associated with epilepsy, is epilepsy changes the voltage sensitivity of the channel. And so many of these change what is called voltage dependence of activation, or the kinetics of inactivation, and it makes the channel open a little more quickly in response to depolarization of the membrane. The same is true for epileptic encephalopathy. Actually, we identify many, many more of these because it's much more severe and the children get exome sequenced. Some of these are larger dots, and that actually is because they pop up in more than one child. But again, you see them associated with the fourth transmembrane domain in a lot of regions, and also the C-terminus. So this is consistent with the idea of epilepsies changing the voltage sensitivity of the channel. On the flip side, those associated with autism are not missense variants, or there's a predominance of missense variants, but it's something called a protein truncating variant, where it introduces a stop codon or some sort of frame shift where the channel is actually no longer made. And so once translation occurs and actually gets to one of these points, you basically have that small product, and that leads to nonsense-mediated decay. The rest of the channel is not made. And so all of these dots, even though they're popping up on different parts of the channel, are equivalent in that they destroy channel function by destroying the protein. What was kind of strange was that it wasn't just protein truncating variants in autism. There actually were some missense variants, and these are more commonly associated with epilepsy. What was interesting was these missense variants are not popping up in the same place as those associated with early epilepsy. In fact, they seem to be associated more commonly with the N-terminus, or very, very close to the ion selectivity filter. We found these repeats here of this arginine very, very close. And what we did first was simply ask the question of what do these missense variants do? And so we did that in heterologous systems where you introduce these into introduce these wild type or these variant channels into HEK cells and just look at the voltage dependence. This is what a normal sodium channel looks like in terms of its inward currents. And this is one where, interestingly, this is a missense variant at one of those arginines, but you can see that the channel, even though the channel actually is at the membrane, as far as immunohistochemistry will tell you, there's actually no current. And so this was a very common feature amongst many of these ASD-associated variants, even those that were missense, is that we basically got absolutely no current out of them. Every once in a while, we got a few that produced a little bit of current. And so what we did then was take those and look at the voltage sensitivity, look at their kinetics, do a little bit of noise analysis to look at conductance, and also see whether or not they got to the surface. Take all of that information and put that into a compartmental model of a pyramidal cell to see what the prediction would be in terms of once you take a channel that has slightly altered kinetics or voltage dependence and instantiate it into a channel that has NAV 1.2, or instantiate it into a cell that has NAV 1.2 channels, what happens to the activity of that cell? So under normal conditions, you inject a little bit of current into this model and you get a few spikes. What we did was first do some validation that the model is replicating what we would expect with epilepsy. And so we instantiated some of the epilepsy variants, and those indeed gave rise to higher activities, higher spike generation or higher frequency. But then if we took all of these ASD-associated variants, what we found is that they either they basically blunted activity. And so we're left with this model where there's this yin-yang association with the known epilepsy variants and the known ASD variants, where compared to wild type levels, we either have a gain of function or a loss of function in terms of spike generation. And so that was our principal general model associated with how one gene can give rise to these multiple disorders. Under normal conditions, you have normal voltage dependence and normal activity. If you reduce it, it can give rise to autism. If you increase it, you have different degrees of seizure. And what I didn't show you today is that other people have established extremely well that the severity of the shifts in voltage dependence or in kinetics can give rise to either forms of seizure that resolve with age or forms of seizure that never resolve with age. Okay, so at that point, we wanted to ask this question of what is going on in, once we have this case where we have loss of function, what is actually going on within neurons? What is going on within cells? Or more accurately question, how does this dysfunction affect neuronal integration? We can understand how gain of function increases the activity of neurons quite easily, but we don't know what loss of function is going to be doing. And so we wanted to ask that question and actually start to study some part of the brain in which SCN2A is important. The first thing you have to ask yourself is, well, what part of the brain do you want to study? The world is your oyster, or the brain is your oyster as far as SCN2A is concerned. It's pretty much expressed everywhere. You can see that we have some very heavy expression in cerebellum. Here it's expressed exclusively in granule cells. You can see that where if you look at the parallel fibers up in the molecular layer, you can see it's NAV 1.2 co-expresses with V-GLUT1, which is in those granule cell parallel fibers. You can see it perfectly in there. We are now starting to study this. This is a project from Chenyu Wang in the lab. And he's just built his own OKR VOR rig. And we're kind of really excited to see processes of cerebellar plasticity that are interrupted in cases where potentially you have loss or altered exponential propagation in these axons. You can also study it in the hippocampus. And Tom McHugh's lab has done some really nice in vivo studies showing that SCN2A loss there impairs sharp wave ripples and also maze navigation. And you can see here, again, it is co-localized with the parallel fibers in these unmyelinated pyramidal cells. You can see that it's, again, perfectly co-localized with VGLUT1. This is Andrea Lorentz's work with Zoltan Nusser, where they showed that the dendrites are studied with NAB 1.6, but not 1.2. But then also you could study it in the neocortex, and this is one of my favorite places to do some research these days. In particular, we're focused on the prefrontal cortex because this is an area associated or strongly associated with dysfunction in autism spectrum disorder. And the neocortex has a few advantages over these other regions in terms of studying sodium channels. And one of them is that we have this very interesting developmental program that occurs within neocortex in terms of the distribution of sodium channels over development. And so really early in development, NAB 1.2 channels, shown by Steve Petru's lab, are all along the axon initial segment. And this is the axon initial segment shown with its cytoskeletal marker anchorin G. You can see that NAB 1.2 basically captures the entire initial segment at this age. This is at postnatal day 3 in a mouse. Later in development, something interesting happens, shown by Yosheng Shu's lab, where if you look at the anchorin G staining and you compare that to NAB 1.2, it's actually only in the proximal initial segment. And that's because there's another sodium channel that takes on a mature role in action potential initiation in that region. So to schematize that a little better, in a developing neuron, both the proximal and distal initial segments are known to express NAB 1.2 early in life. And then there's this shift at about a week in life in a mouse, but about a year in life in a human, where later on SCN2A, which has its own cadre of early childhood epilepsies associated with it, also autism spectrum disorder, it encodes NAB 1.6. It basically kicks NAB 1.2 out of the distal initial segment and relegates it to the proximal initial segment in a mature neuron. And since this channel has a slightly different voltage dependence and it's more hyperpolarized in terms of its activation and its inactivation, it takes on this role of firing action potentials. And when those action potentials fire, they propagate forward, but then they also propagate backwards into the soma, and that's called back propagation. And what people think that NAB 1.2 is there to do in this proximal initial segment is to serve as this reserve pool of sodium channels to help boost the action potential as it encounters this very large soma. And so it corrects for this impedance mismatch that's found in this region to help provide reliable back propagation in this region. And so here we have basically two different functions of sodium channels, and two different predictions for what could happen to the physiology of these cells if you lose some of your sodium channels. So for example, in autism, where you have haploinsufficiency, you've lost half of your sodium channels because one of them has, one of your alleles is now protein truncated or non-conducting. You have 50% of your sodium channels in the initial segment and all of the initial segment. And so here you would predict that you would lose spike initiation entirely. Here, since in mature cells, since SCN2A is taking that role of spike initiation, you actually wouldn't expect that you would have any changes in the excitability of the cell, but you might have changes in your ability to sort of charge this somatic region. So this is something we wanted to look at. And the way we looked at that first, this is Perry and Roy's work, is to just patch cells at various ages and inject current and see what kind of spikes they fire. So here's a developing neuron patched, visualized under a 2-photon microscope, and you inject a little bit of current because these are super tight cells at this age. And you can see in a wild type cell, you generate a good number of action potentials, but in a heterozygote, you actually start to piddle out, you start to peter out in terms of the number of action potentials that are generated. And you can see that the firing curve per current injected is much lower in a heterozygote SCN2A animal compared to its littermate brethren. And this is all stuff that's published last year. Just as we predicted, if you go into more mature cells, you get a recovery of this spike excitability. In fact, at some, I mean, you can start to see that it teases apart. And in fact, if anything, the SCN2A heterozygotes are starting to become a little more excitable than the wild type littermate recordings. Okay, so this is consistent. Oh, sorry. We looked at this all throughout age, and one of the things we did was look at spike threshold as a function of age, and this is on a log scale from postnatal day 4 to 64, where very early in life we see a difference in the spike threshold by about 7 millivolts, but this quickly resolves to the point where these curves converge right around this age between P7 and P10, and that's very consistent with this idea that in mouse, at about a week in life, SCN2A expression is increasing and taking over this role of spike initiation. And so that makes a lot of sense. It also potentially helps explain why benign infantile seizures, which are very mild changes in the voltage dependence of these channels, resolves, because later in life, NAV 1.6 comes online and just takes over the process. And then it doesn't matter whether you have a small change in voltage dependence of 1.2, 1.6 is driving the spikes. Okay, so under these conditions, we have a very early deficit in excitability in neocortex of the pyramidal cells. We wondered how that could give rise to something associated with autism, which is much more associated with either synaptic genes or chromatin modifiers, potentially of synaptic genes, where typically you'll find dysfunction in synaptic transmission, and that dysfunction lasts throughout life. And so we wanted to look at that and see whether or not this early deficit here could give rise to changes in synaptic function. And the first thing we did was look at minis, because that's the simplest and easiest thing to do. And these are spontaneous excitatory events. We also looked at inhibitory events, where these downward deflections are individual release events, and their frequency and amplitude tell you a little bit about potentials for dysfunction in the pre and postsynaptic compartments. What was interesting was at postnatal day 6, we actually had no difference between a wild type and a heterozygote animal in terms of its frequency or its amplitude. And this is the age in which we have this hypoexcitability deficit. What was surprising is that much later in life, at postnatal day 27, way down here, when these curves have converged, we actually noticed a very selective reduction in the frequency. It was about a 50% reduction in the frequency of these events, no change in the amplitude. Now, typically, this change in frequency could be associated with a change in presynaptic release probability. We looked at that using another assay called paired pulse ratio, and we actually didn't see anything associated with that. And so we thought, well, there might be something else going on in terms of synaptic function, and that is a postsynaptic effect. So normally, when excitatory synapses are developing, they start by having almost exclusively NMDA receptors in their postsynaptic density. These are voltage dependent, and they're blocked by magnesium at hyperpolarized potentials. And so when we were doing these recordings, which were AMPA mediated only, we would not pick out a synapse that contained only NMDA receptors. Later in life, as a synapse matures, it starts to incorporate AMPA receptors, and it goes from this case where it's at -80 silent to now being unsilenced. Where it has AMPA receptors, it strengthens, and these are the ones that we would pick up with our mini analysis. And so another possibility was that in the SCN2A heterozygote case, we might have had an excess of NMDA only or NMDA preferring synapses compared to mature synapses that have a complement of both NMDA and AMPA receptors. And so one way to look at that is to do an AMPA to NMDA ratio. I'm showing these flipped for a second, but if you focus on the SCN2A wild type, if you hold the cell at -80, you'd have a nice AMPA component. If you then step the cell to +30 to relieve the magnesium block on the NMDA receptors, you can see there's a mixed AMPA and then slower NMDA component. And this is indicative of a mature synapse where you have an excess of AMPA compared to NMDA. In the SCN2A heterozygote cells, we actually had a much lower AMPA to NMDA ratio, which is consistent with having more of these synapses that appear more silent. And this is the bulk summary across a bunch of different cells. This sort of difference in synapse function has an anatomical correlate. Synapses that are extremely strong tend to have spines that look more mushroomy or stubby, and you can see that they have a large head compared to their neck and compared to the shaft. Those that are more immature, that tend to express only NMDA receptors, have more filopodial processes. And so if we have an excess of silent synapse, we would also expect an excess of filopodial type synapses if we look at them under our 2-photon microscope. That's indeed what we found in these SCN2A heterozygate animals, especially if you look in the apical dendrites. You'll find that they're much more, it's much more common to find these immature filopodial synapses compared to SCN2A conditions. Okay, so at this point, we have this conundrum. We have these deficits that are arising in overall excitability of the cell early, and then synaptic deficits that are arising much later. Something is mismatched here. Either the prediction is that we have this early deficit that's giving rise to long-lasting deficits in synaptic function, or there's something else about the function of SCN2A later in life that we simply have missed with this sort of analysis. And so we decided to look at the exponentials a little more carefully to see if there are other hallmarks, especially considering that role in terms of backpropagation of and charging of the soma that the mature SCN2A channels are supposed to be doing in the proximal initial segment. And so we decided to look at the exponentials a little bit differently, and that is to look, instead of looking at their voltage as a function of time, let's look at the speed at which the voltage is changing as a function of time. And that's something called a phase plane plot, or a guitar plot, as some people like to call them, where you can see that they're during the rising phase of an action potential, and here there's just a doublet to show you different spikes. During the rising phase, there's actually two different components of the rise, and then you get to the peak of the action potential here, and then repolarization. This is spike threshold right here. These have very important these actually reflect activity within a cell. So here is spike threshold. This first hump within the phase plane plot is actually the initiation of the action potential that occurs out in the distal axon initial segment, and you're picking out the cytosolic backpropagation of charge into your pipette. Eventually, that will then depolarize and activate somatic sodium channels and gives rise to the second hump, which is associated with the influx of sodium right at the somatic membrane. And you can see that these humps sort of tell you with a little more nuance about what's going on in different compartments of the cell. And you just can't pick those out when you look at an action potential under normal conditions. And so we want to look at this sort of assay as a function of age. We can see that develops over time, and it develops in a consistent way in a wild type animal, where when you start with really depolarized threshold, you actually have very slow spikes because you just don't have the cell's just not mature yet. But eventually the threshold hyperpolarizes and the spikes get faster and faster and faster. And when the threshold plateaus at about -54, the spike speed also plateaus at a peak of about 580 volts per second. What was interesting is if we look at the SCN2A heterozygate animals, we noticed a deficit in the peak rising speed of the action potential that actually increased in terms of its deficit over age. And so even though there wasn't much difference in the spike speed early in life, there was actually a very massive, about 30% reduction in spike speed later in life. And we wondered what effect that could have on cell function. What was interesting is this occurred basically all throughout cortex, neocortex, but didn't occur in hippocampus. And that's consistent with this idea of NAV 1.6 having a predominant role in the soma and dendrites of these cells, whereas maybe 1.2 is having a different role in the soma and dendrites of neocortical cells. And so we want to ask the question, well, what are the conditions that could give rise to this deficit, this model? And so the original model that we started out with was NAV 1.2 in the proximal initial segment. And then based on this hippocampal knowledge, 1.6 basically distributed everywhere. If you do that and you look at spikes in a model, you actually see that the initial segment hump is reduced, but that's not recapitulating what we see empirically. Instead, what we had to do was incorporate an equal number of 1.2 and 1.6 channels in the soma and the dendrites, and under those conditions, we see a pretty good recapitulation of what's going on in the experimental data. What's interesting at that point is that with this, you have this prediction. If you have 1.2 out in the dendrites and in the soma, if you lose half of them, you have this about halfway reduction in spike speed. If you lose all of them, you should have a larger reduction in spike speed. And now we weren't able to do this in these heterozygote animals because if you generate a homozygote, they unfortunately are perinatal lethal because they can't drive their diaphragm. But you can develop conditional animals where you completely get rid of NAV 1.2. And if we do that in double flox mice, we actually do see that deficit consistent with this model. And so this helps validate our model, but it also opens up a possibility to study what happens when in excitability when you completely get rid of NAV 1.2 in these cells. If we look at that across a bunch of cells, we actually see that there's a pretty marked change in the peak speed of the spike in the soma. Interestingly, there's a little hyperpolarization in threshold, but it's on the order of 0.8 millivolts, so it's really not that severe in terms of the average. So it seems like the most marked effect is happening in the soma. What was surprising then was to find that these cells, instead of being hypo-excitable, which is what you would predict from losing a sodium channel, actually become hyper-excitable. And this is kind of interesting because, as I mentioned, some of those kids that have a loss of function in their sodium channel actually develop seizure as well. And so we wondered whether or not this full loss of SCN2A might be able to tell us something about the mechanism. And so we looked at that over a population. What we're seeing is that the cells seem to be a little more bursty. And their FI curve is dramatically raised. Now that we've added many more heterozygote cells, you can see that the heterozygote curve is starting to peel away from the wild type curve as well, indicative of potentially a little more excitability even in this heterozygote case, which would be consistent with the childhood phenotype. We wondered, why is this happening? Is it compensation from another sodium channel, maybe 1.6? Is there something else going on? Basically, we've been looking at everything. And to make a long story short, we see no real evidence of compensation. There's a little bit of upregulation of 1.6, but it can't explain this hyperexcitability. Instead, what we're finding is something kind of interesting, and that is that basically the spike is not as large. And since the spike is not as large and doesn't depolarize as much as it should, it's not driving potassium channels as well as it should. So this is in a model where we see the sodium charge is going down and down and down. That actually leads to less engagement of potassium channels. And so basically the hyperpolarization after these action potentials has been reduced. If we look at the charge ratio between sodium and potassium in these models, you can see under wild type cases, you have this value. Under heterozygote cases, you have a little bit of a change, consistent with a little bit of a change in excitability, but then you really fall off a cliff when you have a full knockout. In the model, all you have to do is just completely remove NAV 1.2, and you can see that the cells don't hyperpolarize as well, and then it gets back to firing a new spike. Granted, that new spike is not as high or as large in the soma as what you normally get, but as far as forward propagation counts, that doesn't matter because that's all mediated by 1.6. So once you fire off something, then it becomes an all or none action potential going down the axon. And as far as overall action potential excitability is concerned, these cells are very hyper-excitable, which looks to be due to a case of not engaging these potassium channels and not hyperpolarizing the cell between spikes as well. And we're starting to see evidence of that in in our recordings, where the spike height is just not as high, but the cell doesn't hyperpolarize as much, and so you just get this rebound, and it's much more likely to fire excess spikes. And so it's this somewhat counterintuitive effect where losing a sodium channel makes a cell hyper-excitable, but it's because of its ability to engage potassium channels. And this is reminiscent of work from Bernardo Rudi's lab, where they showed that fast spiking is due to strong hyperpolarization via potassium channels that allows you to deactivate sodium channels to fire the next spike. And so this might be consistent with this sort of idea, and it might be consistent with why some children are generating seizure, because maybe other factors are pushing or pulling them in various directions, and some children might be closer to falling off this cliff in the heterozygote case. Some other variables that are found in their cells. Okay, so with that little aside, let's get back to this idea of haploinsufficiency and what it's affecting. So now that we've seen this deficit in the soma, it actually has an effect that we would predict would be much more severe as you go back and back and back into the cell. And if you look in the model in terms of backpropagation of that ax potential throughout the dendrite, you can see that the ax potential gets smaller and smaller. These backpropagating ax potentials are really important for synaptic function and synaptic plasticity and potentially maintenance of synaptic function, because they provide an instructive cue for these synapses in terms of what's going on at the soma, and serve as a coincidence detector for coincident activity between incoming synaptic activity at one point in the dendrite and then ax potential output at the soma. And so we wondered whether or not this was really impaired, and the way to do that would be to look at, not at sodium imaging, because it's really, really hard to do that with the sodium dyes that are available today, but actually look at calcium imaging, and that's because sodium channels depolarize cells, that depolarizes calcium channels have beautiful indicators, and then you can get nice calcium influx in various regions of the cell. So what we did was look at backpropagation of action potentials, and here we looked at backpropagation of bursts. You can see that under normal conditions, we get this robust engagement of calcium channels all throughout the dendrites of these cells. In a heterozygote case, we actually saw that was very severely attenuated. And this was attenuated also in a wild type case where we added just a little bit of TTX to mimic the SCN2A heterozygote conditions. So this suggests that the function of NAV1.2 is occurring throughout life. And there's an online active form of NAV1.2 in these dendrites that's giving rise to this deficit in backpropagation. It's not some developmental effect. This, as I mentioned, is important for synaptic function and plasticity. And one thing that was interesting is if we take that same protocol and incorporate it into a burst-dependent, spike timing-dependent plasticity protocol, where we activate these synapses in layer 1 and pair it with backpropagating spikes. Under normal conditions, we get pretty nice LTP, about 50%. In the heterozygote case, we see absolutely nothing in terms of LTP engagement at those synapses. And again, it's mimicked by adding a little bit of TTX to a wild type cell. And so that suggests that we have this change over development in NAV1.2 function that we were able to learn basically from looking at constitutive animals, constitutive SCN2A loss animals, where early in life we have impaired ax potential initiation. Later in life, we have this impaired dendritic excitability. And also later in life, we have this impairment in synapses, impairment in synaptic plasticity that we think is associated with this deficit in backpropagation. But honestly, we haven't proven that at this point, because we've been looking at animals that have gone through life with both of these deficits. And so the next thing we wanted to do was disentangle these two processes. And that would be, instead of using a constitutive animal, to use a conditional animal where we can convert them from a wild type into a heterozygote basically any time in life. And so we did it where we allowed the animals to grow up with normal excitability, normal ax potential initiation early in life, and then induce heterozygosity about here. And so what you would expect is that the animals would go along this curve and then sort of fall off this curve in terms of excitability. We did this by crossing it to a CAMK-CRE driver line, and that has the advantage of, one, being expressed pretty much in excitatory cells in neocortex, and 2, the CRI actually kicks on late in life at about postnatal day 16 or so. So we can basically have this case where we take all the pyramidal cells and convert them from wild type into heterozygotes at that point. That's indeed what we see in terms of spike speed. And interestingly, when we do that, we can see that backpropagation is impaired in these cases. We can also do it a slightly different way. Instead of doing it in sort of neocortex pyramidal cell wide, we can do it in a cell autonomous way. We inject a little bit of CRI, and this we did at postnatal day 28, and we'd see CRI positive and CRI negative cells, patch onto those next to each other, and we'd see that the positive cells look like constitutive heterozygotes, the negative cells look like wild type cells. And so this suggests that there's a cell autonomous effect here. And then we wanted to ask, well, of course you would expect a change in spike speed when you get rid of sodium channels, but what happens to the synapses? Is the synapse function due to this deficit in spike speed, deficit in backpropagation, or deficit in dendritic excitability? This is our deficit in terms of AMPA to NMDA ratio in the constitutive case. And then if we look in these 2 different conditional cases, we actually see a very consistent effect where we see about, again, a 50% reduction in AMPA to NMDA ratio, independent of which way you destroy sodium channels or you remove sodium channels. And so this suggests that basically at any age, you can get a reduction in spike speed, and then you can get a reduction in synapse function. It suggests that NAV1.2's function is to actively maintain the synapse strength over development, or throughout development, throughout life. Okay? And so that opened up the question of, well, if we can get rid of it anytime we want, could we actually restore it at some point? And this opens up the possibility that we might be able to develop a therapy for children. And so one way we wanted to look at that first is, well, can we develop sort of the gold standard of what could be a therapy? And that is to go from a case where you have heterozygosity back to completely normal, both alleles functioning, NAV1.2 function. So instead of using a conditional knockout, we generated a conditional knock-in where under the flipped condition where we flipped exons 3 through 5, we actually knocked in also some GFP. So all of the heterozygote cells under baseline conditions should be GFP positive. And then with pre-recombination, we can knock the GFP out, but then flip back exons 3 through 5 and go to a wild type condition. We actually see that under our 2-photon. Here are a bunch of GFP positive pyramidal cells in this animal, and every once in a while you see a negative cell, for example, here, here, here. These look more like FSIs, like interneurons that should not express NAV1.2, so we need to examine that a little bit further. But now we can ask the question of, well, what happens when you restore NAV1.2 basically any time in life? Well, the other thing we needed to do was validate that this animal is working. So under heterozygote conditions, baseline conditions, we see the spike speed is down. It looks just like a constitutive heterozygote. But with an injection at postnatal day 30, pretty much peri-adolescent in a mouse, we can see a restoration in action potential velocity. We also see a restoration in backpropagation. So this is the CRI negative in red is the SCN2A heterozygote case. If we reintroduce and turn it back into a wild type, we see a recovery in backpropagation that looks a lot like our constitutive animal. But then the real question is, what happens to the synapses? So again, we looked at AMPA to NMDA ratio, which was down in the baseline case. But once you restore NAV1.2, even though you restored it very, very late, you have an increase in AMPA to NMDA ratio, which opens up the possibility that you might be able to get some recovery in a child if you could in some way restore NAV1.2 function. And that's the last question I wanted to talk about today is, could we develop something that looks like a therapy for these families? And so this is something where you start to think about pharmacological manipulations. Unfortunately, it's very, very hard to increase conductance through sodium channels without changing the voltage dependence, without changing the kinetics. And that, as we all know, actually gives rise more easily to epilepsy than a treatment for autism. You think about gene therapy. Unfortunately, NAV1.2 or SCN2A is just too large to package into a virus for traditional gene therapy. And so we had to think about different approaches. And one of those is has been developed quite well by Nadav Hituv's lab just across the street from us, where they're working on CRISPR technology using CRISPR activators. And so under CRISPR activation, what you have is you have a gene and you have some regulatory element upstream. What you do, or what they do, because I don't know how this actually works at the molecular level, I'm a squiggly line guy. You take a nuclease deficient version of Cas9, so it doesn't make cuts in the DNA, but it actually serves as a chaperone for something else to whatever part of DNA you care about. And in this case, it's the regulatory element. And you pair it with some sort of effector. And that effector can either repress transcription, that's called CRISPR inhibition, or it can overdrive transcription, and that's called CRISPR activation or CRISPR-A. This is the form that we want to do, because what we have is we have one normal allele, one protein truncated allele, and what we want to do is overdrive that normal allele to rescue or to compensate for the lack of expression on the protein truncated allele. So Nadav's lab is focused on this, and then Navneet Matharu has done this beautiful work in animals that are haploinsufficient for this gene called SIM1, which gives rise, in the haploinsufficient case, to obesity. And you can see that under SIM1 conditions, these animals are pretty porky. The wild types look normal. What they did is they did a single injection of a CRISPR activator of SIM1 into the hypothalamus of these animals. And you can see that these heterozygotes had rescued the weight of a wild type littermate. And so this they published last year in Science, and we're now working with them to translate this over to SCN2A. There are a number of other advantages in terms of sort of gene therapy here. One, there's no actual DNA editing. The CRISPR is serving as a chaperone. It's not making cuts, so you don't have to worry about off-target cuts. It should work for all protein truncating variants, regardless of where that variant is. So you don't have to design a specific gene therapy for a specific mutation or variant that's found in the channel. It takes advantage of all the endogenous regulatory machinery. And so they showed quite nicely with SIM1 that the upregulation is occurring just in cells that express SIM1. The same should be true with SCN2A. And in theory, a single administration should be effective for life because they're non-dividing cells in the brain. And so if we can get it into the brain and upregulate SCN2A, we might be able to restore some of these functions. And so this is the work of Serena Tamura, who's been working to design plasmids that target different promoters or enhancer elements upstream of SCN2A. She found a few when you look at upregulation in Neuro 2A cells. We packaged that into AAVs, and one of them works pretty darn well. So we then packaged that into a DJ construct. I should mention, actually, we're doing this in a split virus process right now, because these are still a little too large if you want to package a fluorescent reporter. And so we have the guide RNA in one plasmid, and then the Cas9 and the transcriptional activator BP64 in another. But if we do that and we inject those together into the prefrontal cortex, we can see an upregulation in SCN2A in both wild type and heterozygote cases. And we see a rescue in spike speed under these cases, which actually is pretty darn good. It's not 100%, but we're getting there. Some of these might be false negatives or false positives where we have M. cherry, but we don't actually have both viruses in there. But what's more interesting is even though we have a partial recovery in spike speed, we actually get a really pronounced recovery in AMPA to NMDA ratio, which again is consistent with this idea of if you can restore some of the function of NAV 1.2, you can have some benefit for these synapses. And what we're now starting to ask is whether or not we can have a benefit in basically in the entire animal. But that requires you to inject basically the whole and get brain-wide penetrance. And so we're exploring other viral approaches to do that. Now, one concern with this is that you don't want to have a case where you have a gene therapy that rescues too far. Because if you rescue too far, you go from autism spectrum to some form of severe epilepsy. And so we really worried about that. And we wondered whether or not in the wild type cell, if we overexpress NAV 1.2, or overexpress SCN2A using this CRISPR construct, whether we have any change in excitability that would suggest that we have hyper-excitability. One thing that we found that was kind of surprising was that we actually didn't. So we see that the overexpression in a wild type doesn't make the cells even more excitable than under wild type conditions. So it suggests that there's some sort of ceiling effect here, whether it be these alpha subunits of the sodium channels need to be co-packaged with beta subunit isoforms to have membrane insertion and membrane function, and we're not affecting the beta subunits, or there are a number of slots in the membrane that are available for sodium channels that are already filled. Whatever it is, we're not seeing overexpression lead to hyperexcitability, which suggests that we actually have a pretty large therapeutic window if we're able to overexpress under these cases. We might not have a ceiling effect. So that's really, really enticing for the future. Okay, so at that point, I have run out of time, I think. I just want to cap by saying, by giving you a quick overview of the story so far. Basically, what we see in Neocortex is that 1.2 repositions from the axon to the dendrite during development. I mentioned briefly that that story is different depending on whether or not the cell is myelinated or unmyelinated, and you have a different functional 1.2 in other brain regions. So if we really want to understand what it does brain-wide, we have to study those other regions. We're starting to do that. But in Neocortex, it looks like this dendritic activity is really crucial for excitatory synapse function. We still have to figure out what's going on with that early axonal deficit, whether or not that affects the maturation of the cortex. We haven't seen anything so far, but we can't rule out anything. What's really exciting is that this developmental rescue, using whatever tool you want, is enough to rescue some aspects of AP waveform and synapse strength. What we now are working to do is see whether or not that translates into behavioral rescue in these animals. But it opens up the possibility that if we have this cis regulation therapy, which is CRISPR-A, it opens up the possibility that it has therapeutic potential for these children, and there might be an extended developmental window in which we could administer this to see some therapeutic benefit. What's also interesting is this opens up the possibility for cis regulation therapy in a range of ASD-associated genes, because many of these genes are unfortunately too large for traditional gene therapy, and this sort of approach where you target the enhancers or promoters might be much more beneficial. Okay, so with that, I want to thank the team and thank the funding. This has been supported almost entirely by the Safari Foundation, with also a lot of support from the Broad Institute, the Family Foundation. The rest of the lab is supported by a bunch of different sources, including the NIH, and Perry was supported by the Canadian government. And as I mentioned, Roy and Perry are principal drivers, and Serena, principal drivers of this program. We have some new additions to the lab, including Chen-Yu Wang, who's working on Cerebellum, Andrew Nelson, who's working on interactions between sodium channels and its scaffolding proteins in development. And this is done with a lot of help from local collaborators, including Stefan Sanders, Nadav Hituv, Evan Feinberg, Massimo Scanziani's lab, and Dev Manoli's lab. So with that, I want to thank everybody for their attention this morning, this afternoon, this evening, the middle of the night, and hope you're all doing well, staying safe. And I guess I'll stop sharing my screen.
I can also close, so that's fine.
Okay.
Thank you very much, Kevin. Clapping for everyone. That was a really nice and comprehensive story. I start reading. We have one question. Don't be shy. So for the others, you can write your question at the bottom of the screen on the Ask a Question option. So I start reading the first one. So hello, great talk. First, I have two questions. First, they ask whether did the conditional animal also show immature spines, which I guess, I don't know if that refers to the CAMK animal.
Yeah, so we looked at that a little bit, but we haven't gone through and analyzed completely. Actually, no, we did. And actually, they don't have a severe deficit. And so there seems to be a bit of a disconnect there in terms of synapse function. And what might be happening is those synapses are actually growing into mushrooms, and then they're starting to regress into something else. What's interesting there is that we see this change in AMPA to NMDA ratio in those animals, but also if you look at many changes in minis, we don't see a real overt change in frequency at that point. We actually see a reduction in amplitude, which is consistent with this idea of the silent synapses have grown into mature synapses and then are starting to lose a few AMPA receptors. And so their AMPA component amplitude has gone down, but they don't convert back into completely silent synapses. And yet the ratio of AMPA to NMDA does change. And so there is this interesting developmental window of, if you grow up with silent synapses, you appear to maintain silent synapses in the heterozygotes. But if you conditionally start as a wild type, are able to grow mature synapses and then have them regress, there's something else going on. And we saw a small change in spine morphology, but I don't think it was significant. Now, in terms of the behavior, oh, that was the second part of that, right?
Yes, that's the second part. And I mean, I think, yeah, I wanted to add to that, you touch upon at the end and the conclusion. I mean, it seems the perfect case for you now to disentangle what is the function of the very early phase of development, when you see this actual potential difference from what is the impact on behavior and what is the late or later phase impact on behavior. And I guess this is kind of hint also the second question, what about the behavior of the Cree animals versus
well, we actually don't. We haven't studied the behavior of the Cree animals because the behavior of the constitutive animals was so slight in terms of the difference that we've looked at. We did this sort of standard ASD panel of looking at overall mobility, anxiety, all sorts of things. So that's summarized in Perry's paper in the supplementals. The long and the short of it is there was a slight effect in terms of social memory preference or social in the females, and then in the males there was a little bit of a deficit in maze navigation, or and that's consistent with what Tom McHugh's lab has seen in their males in H-maze navigation. But it seems like you actually have to tax these animals quite hard to be able to see some differences, which is kind of interesting.
But because it's very connected to these, I have a question, and namely, do you know whether the expression of the SCN2A is similar also in terms of time frame in human neurons?
Yeah, we think so. So the trick is that it's hard to get tissue and do the staining, of course. But when it has been done, it's sort of this consistent effect. And actually, you see it more easily with gene expression studies, where you see a developmental trajectory. And this idea where in a mouse there is this nice switch at about a week in life, that is very consistent with the developmental trajectory that you see in humans that occurs about a year or two in life. And so we think that these are consistent. When people have stained for sodium channels in adult postmortem tissue in human, they see a very similar distribution in proximal initial segment for 1.2, distal initial segment, and the nodes in human neocortex. Yeah.
So I go to the next question, very quick. Are the humans without this mutation homo or heterozygous?
Oh, sorry, I should have clarified that. So these are invariably heterozygote cases where you have one functional allele and one altered allele. What we're talking about today, all of those genes that we talked about are de novo mutations. These are the really highly penetrant mutations that are the ones that we can study, where usually it gives rise to protein truncation. So you're in a haploinsufficiency case.
So nice question. I think you already partially answered to that. I don't know if you want to add something. Let's say beautiful talk first. Do SCN2A CAMK2 Cree or F1A Cree condition knockout mice also show decreased AMPA and NMDA ratio, or on the opposite way, just like the cell excitability? And is it the homeostasis that regulates excitatory synapse development after loss of SCN2A?
Yeah, so The first answer is yes. So if you reduce excitability, you get a reduction in AMPA to NMDA ratio. If you restore excitability, you get a restoration in AMPA to NMDA ratio. Is it homeostasis that regulates excitatory synapse atrophy? Depends on what your definition of homeostasis is, of course. The way I like to think about it is it seems to be something to do with overall dendritic excitability. Now, we focused on backpropagation because it's a relatively easy thing to study. One of the things that we're moving towards now is that, honestly, backpropagation is just one aspect of dendritic excitability. There's also branch-specific excitability where you can get the interaction between different synaptic inputs within an individual branch give rise to local dendritic activity, engagement of NMDA receptors, engagement of calcium channels. That sort of excitability also seems to be impaired in the SCN2A heterozygate cases. Very preliminary data there. And so is that loss something that's homeostatic? I don't really know how to answer. I'm not really a homeostasis expert. But certainly there is a set point, and we're in terms of excitability, and we're not there in that set point.
So I read the last two questions. So the first one, do you think any of the variability in symptoms could come from change in interaction with absolute units?
Yes, definitely. So some of these missense mutations, especially those that are on the tails, could have interactions with beta subunits, and the beta subunits themselves have their own slew of ASD or autism associations. And so it's an incredibly complex problem when you start to think that there are auxiliary subunits that are just the beta subunits, because those will change the voltage dependence of the channel. But also another thing to consider is that there's also scaffolding proteins, for example, the anchorins, and any interactions with the anchorins that are interfered with could also give rise to some deficits. One thing that's nested in here is that there is variability at the biophysical level in terms of how an individual variant associated with ASD affects the channel, and then that affects a child. And you would hope that there would be a phenotype-genotype correlation there. We looked at this a little bit in the Ben Shalom paper. Unfortunately, we basically have an N of 7 where there's deep phenotyping, and you have deep biophysical characterization. And at that level, we can't say heads or tails about what's going on. The hope would be this is the type of channel, this is the type of protein where you could start to do that once you have a large enough cohort. Yes, so that's a really interesting question.
With all those cases, it should be possible in that and SCN2A, then those are the...
Yeah.
Last question. I didn't get if excitability deficit is pre or post-synaptic in relation to the synaptic while you see the NMDA ratio.
Sorry, I went over that part really, really quick. So it looks like it's a post-synaptic effect as far as we can tell. What we found is that the mini frequency is reduced, and that could be due to a reduction in release probability. That didn't appear to be the case because we looked at paired pulse ratio, and that looked to be normal. Now, of course, there are cases where paired pulse ratio is not the best of indicators for changes in release probability, but it looked like it was pretty good under this case. Here, what we found is that on the postsynaptic side, we seem to find that many of the synapses were lacking AMPA receptors, and that was giving rise to the reduction in mini frequency, AMPA mini frequency. And that's consistent with a reduction in AMPA to NMDA ratio. So as far as we can tell, it seems to be a purely postsynaptic effect, which kind of makes sense in neocortex because these excitatory synapses on the presynaptic side are expressing NAV 1.6 in their boutons, and that's supporting exponential propagation down the axon, whereas 1.2 seems to have this role in the dendrites. That's relatively new, but seems to be pretty conclusive as far as we can tell.
Good. So I think we finish here also because we are a bit over time.
Sorry about that.
No, it was perfect. I mean, the nice thing of those platforms is that we can go at as much as we want in principle. So I thank you very much for everyone, and I think stay safe from many sides, from the fires and from the COVID.
I'll be out there in my mask and my hose, and we'll do our best.
Yeah.
Thank you, Gaia, for hosting. Really, really appreciate it. This was.
Thank you for being here.
Thank you, everybody, for the questions and for tuning in.
Yeah, and for everyone else, just remember that. So we started again, and we have a full schedule. I'm going to update it later. Also, on the, you can see in the worldwide website. We have a full schedule till the end of the year. Very exciting talks. So stay there every week on Tuesday at 5 p.m. Vienna time. So see you, and thank you again. Bye.
Okay. Thank you very much. Bye-bye.